All right. Afternoonish, everybody. So we're going to get started. A couple quick reminders as we get going today. First off, there is a quiz today that opens up after class. So there is a quiz on Brightspace, quiz number 5. That'll open up after class. Same format as the quizzes throughout the semester, but it's got 15 points worth of questions, a mixture of multiple choice questions, as well as maybe a short written response question or two then as well. That'll cover, in essence, the last kind of week's worth of materials. It does focus in mostly on the control of growth lecture set, a little bit of today's lecture set, which is going to be about energetics and energy. And then it does actually also have some more population growth math problems on it then as well. And that is because typically, again, those are going to be on the next exam. And it's one of the things that sometimes some of the students in the class struggle a little bit more. So I'm trying to give you guys more practice problems to work through those then. That quiz will stay open until 5 p.m. tomorrow on Thursday the 13th then. The other thing to keep in mind then is that you have an interactive notes set due on Friday. That is going to be the lecture set on microbial growth. That is due on Friday to a Dropbox on Brightspace then. Questions about either one of those two things? Okay, we're going to be lecturing today, lecturing on Friday. Next week is spring break, so obviously we don't have class. Remember, though, we also pushed the exam back. I was trying to get it away from the anatomy exams this week a little bit. I was trying to give you guys some more time to study and prep for exam number 2. But that means you guys should be working over spring break and studying for that. The exam 2 is now going to be on then Wednesday, March 26th. So the Wednesday that we come back from spring break then. The exam is going to be in the same format as the first exam. It's going to be a mixture of some multiple choice questions. It's going to be the pathogens of interest are going to be on there. I will send out a pathogen of interest study guide. I'll get that sent out and posted to Brightspace after class today. This pathogen of interest study guide is just all the pathogens of interest that are on exam 2, just in one spot for you guys to work through. Then it will have a short essay question or two at the end, just like the previous exam did. Questions about that? All right. So today we're going to talk a little bit about enzymes and energetics, and this lecture set is then the last of the exam 2 content then as well. So this lecture set is on exam 2. Once we finish up this particular lecture set, then we're going to move on to exam 3 material. So Friday's class, Monday's class after spring break, I'm going to be covering exam 3 material. And then, but this is going to be, when we reach the end of this lecture set, it's the end of the exam 2 content then. Okay, so exam 2 is going to include things like motility, the microbial growth lecture set, the control of growth lecture set, and then this particular one. Okay. So when we're starting, we're going to be starting to get into our metabolism unit in this particular part of the class. Metabolism is really just describing all of the chemical reactions that take place inside of cells. So metabolism, basically what's going to be happening is there's going to be some type of nutrients out in the environment. Those nutrients are going to be transported into the cell. Those nutrients then go into various different kinds of metabolic chemical reactions. Some of those metabolic chemical reactions will be processing energy for the cell. The cell will need some energy to do certain things. But some of those chemical reactions are also going to process those nutrients instead of for energy for cellular structure, building up structures inside of the cell. And metabolism is really just going to then result in energy for the cell and growth of the cell then over time. Now, again, metabolism, if I had to define what metabolism means and what that term means, metabolism is the sum of all the chemical reactions inside of a cell or inside of an organism. So all the different chemical reactions taking place inside of these organisms summed together, that's going to be metabolism. Metabolism is just describing the chemical reactions inside of organisms. Now, chemical reaction is going to be a conversion then of reactants, or sometimes also substrates, they're called. You can use the two terms reactants or substrates interchangeably. A reactant is the starting molecules of a chemical reaction. They go through a chemical reaction that then is going to change those reactants in some way into then the products of a chemical reaction. And products is what you get out at the end of then a chemical reaction. So the reactants go in and get converted into the products, which is what is made at the end of a chemical reaction. And we could have a various different chemical reactions. One of the ones that I showed last lecture, for instance, was hydrogen peroxide, could get broken down by the catalase enzyme into things then like water and oxygen gas. Anybody know what the problem with this equation is right now? Yeah, it's not balanced. It should actually be, did I get that right? Yeah, oh, 202, yeah. So it should be balanced instead, for instance, but yeah. Okay. You guys don't have to worry about balancing chemical reactions in this class. That's not what we're going to do. Did I still get that wrong? I feel like, all right, it's fine. All right. So what you're going to have to do is, though, typically with these chemical reactions, the reactants are going to get converted through some type of chemical reaction and then the products. You guys, I wrote catalase here above the arrow. What is catalase an example of then? It's an enzyme. So catalase is an enzyme, and we're going to talk about enzymes today as well, okay, and how they participate in chemical reactions. Now, when it comes to metabolism, when it comes to chemical reactions inside the cells, typically metabolism can be broken down into two subcategories. So the types of chemical metabolic reactions inside the cells can fall into two categories. You can have either catabolic reactions or anabolic reactions. Catabolic reactions, these are going to be types of chemical reactions that break large molecules down into smaller molecules. So they're going to cut molecules apart in some way. They're going to break these larger molecules into smaller molecules, but they then also generate energy intermediates for the cell. So catabolic reactions typically process energy for the cell, harvest some energy for the cell, but then store that energy in the form of energy intermediates that are then made by these catabolic reactions. So they break larger molecules down to the smaller molecules, and then they're basically then going to be generating energy intermediates, which is a way the cell stores energy. Anybody think of an example of an energy intermediate and what I mean by that, though? What might be some ways that cell stores or uses for energy? Sometimes they're also referred to as energy currency for cells. So an example of somebody just said it, an example of an energy intermediate is ATP, for instance. ATP, adenosine triphosphate, would be one example of an energy intermediate, and we're going to talk about that a little bit later here today. Anabolic reactions, so catabolic reactions are going to break large molecules down to smaller molecules, and then they make energy intermediates. Anabolic reactions, these are going to be reactions that take small molecules and put them together in making the larger macromolecules. And when I say macromolecules, what do I mean again? What are examples of macromolecules of the four classes? So it could be carbohydrates, and in this case, they're going to be making more specifically polysaccharide carbohydrates. Could be, what else is macromolecules? Huh? Proteins. They could be making proteins. What else? Lipids and nucleic acids. So when I say these, what is really happening is anabolic reactions make these, synthesize these. So anabolic reactions are going to take small molecules, put them together into larger macromolecules like polysaccharide sugar chains, proteins, lipids, nucleic acids like DNA and RNA, and it makes these, and these are going to make up the biomass of the cell. The other part of this, though, definition for anabolic reactions is they make things like the macromolecules, but they also use up energy, and they use up energy in the form of these energy intermediates. So catabolic reactions are going to make energy intermediates. Those energy intermediates are then going to get used up by anabolic reactions to then make the structures and the macromolecules and the biomass of the cell. So again, the main connection between catabolic and anabolic reactions is then going to be the energy intermediates. The catabolic reactions make energy intermediates. Those are then used up by the anabolic reactions. So metabolism is the sum of all of these different kinds of reactions, but some of the reactions are catabolic, some of them are anabolic. The thing with these reactions is we're also typically going to be concerned with energy and energy processing. And these are governed a lot of times by the laws of thermodynamics. The main law of thermodynamics that we're often worried about with living organisms is the second law of thermodynamics. Basically, the second law of thermodynamics says that no reaction is 100% efficient. In every reaction or in every energy transformation, some of the energy is going to get lost as heat energy to the surroundings. And so this is really what is described in the second law of thermodynamics. When we're talking about biological organisms, that's one of the things we're mostly concerned with. So all the time, we are losing some of our energy and radiating it out as heat to our surroundings. That heat energy that we're losing is energy, again, that we're losing. It's not available to us. And that is just because all these chemical reactions inside of our cells, inside of our body, are not 100% efficient. But all organisms are losing heat energy to surroundings. Bacteria, archaea, plants, us, other kinds of animals, we're always constantly losing a little bit of energy in the form of heat. In cellular biology, what we're going to be interested in is what is called free energy. This is the energy that the organisms retain and can use. So free energy is the energy that's available to cells and to organisms for work. It's the energy we're not losing as heat to our surroundings. That the heat energy that we retain is the free energy. And when we have this, some reactions will actually generate free energy. Some reactions use up some of that free energy. So for instance, we can have what is called favorable or spontaneous chemical reactions. Those are typically going to liberate or generate a little bit of free energy. We can have unfavorable or what are called non-spontaneous reactions. These are chemical reactions that require an input of free energy in order to happen, in order for them to take place. And there's ways to describe this. So chemical reactions can also fall into two categories of what are called exergonic reactions or endergonic reactions. And exergonic reactions, this is going to be any chemical reaction that proceeds and over the course of the reaction has a net release of free energy. These are also sometimes, not only are they called exergonic reactions, they're sometimes called also spontaneous reactions or favorable reactions or reactions that have what is called a negative delta G. What's the delta symbol mean in math? Change in. And what this means is that negative delta G has a negative G value, or change in the V value over the course of the reaction. The way to think of then exergonic reactions is really just kind of what's graphed out here. Here on the x-axis is progress of the reaction. So this is going to be the start of the reaction through to the end of the reaction. Again, we're going to start off with reactants, but then we're going to end at the end with products. So again, the reactants are the starting point of the reaction. Products are going to be the ending point of that reaction. With this, the reactants, on the y-axis, this is energy. Again, from maybe 0 energy to a lot of energy, we'll just say. But we're going to go from energetic state from very little energy to higher amounts of energy along the y-axis then. I don't have any units on here, but that's just graphed out, basic graph theory. In an exergonic reaction, the reactants are going to be at a higher energetic state than the products. Anytime the reactants start off at a higher energy state and they're at a higher energy state than the products, every single time, that's a exergonic reaction. So these reactants, let's just say they have 10 units of energy, 10 joules, say. Maybe then our reactants over here are going to be at 3 units of energy. The one thing I didn't go through is the first law of thermodynamics, and that says energy cannot be created or destroyed. So energy just can't disappear. And what happens here is because the reactants are at a higher energy state than the products, one of the products of this chemical reaction is a release of free energy. So over the course of this then reaction, energy gets released or generated. And sometimes the cell can use that energy to do work. Anybody know what the definition of energy is? Okay. Rough definition of energy is the capacity to cause change or to be able to do work. And work is something changing. Okay. Now, what is happening here is you get a net release of free energy. By the way, it says a negative delta G. The way you calculate delta G, the change in energy, is the energy state of the products minus G is the energy state of the reactants. In this particular case, if I wanted to calculate delta G, again, I would have the energy state of the products, which I said was, maybe I just said was 3 if I'm just making it up, minus the energy state of the reactants. Again, I just made it up. It was 10, say. What's 3 minus 10? -7. But I have a negative delta G value. What that means is 7 units of energy are being given off are being released during the course of this reaction. And this is going to make it an exergonic chemical reaction. Questions about that? You can use the term spontaneous reaction or favorable reaction, but these are just alternative ways to say an exergonic reaction. An endergonic reaction is going to be any chemical reaction that instead absorbs energy from its surroundings in order to occur. So an endergonic reaction, the reactants are, say, we have units of 3 energy. Now our products have, say, let's just say they have 10 units of energy. But the reactants are going to start off at a lower energetic state than the products. Anytime the reactants are at a lower energy state than the products, it's an endergonic reaction. Although this is sometimes also called a non-spontaneous reaction or an unfavorable reaction. Those are 3 terms that are used somewhat interchangeably in some cases. It's also said you're going to be having a positive delta G value because, again, delta G is then going to be the energy state of the products. I said that was 10 here in this case, minus the energy state of the reactants. I said there was 3, and 10 minus 3 is positive 7. Does that make sense, everybody? Now, this is an endergonic reaction because for this reaction to occur, energy cannot be created or destroyed. It has to come from somewhere. The reactants are at a lower energy state from the products. So you cannot just suddenly make the reactants of the products unless you put in some energy to make this reaction occur. Endergonic reactions require an input of energy. Endergonic input of energy, NN. Okay? They require energy to be put in from some outside source in order to occur. If you do not have an energy input, that reaction cannot happen. This is also why it's typically called then also a non-spontaneous reaction. If somebody is spontaneous, it doesn't, spontaneous, non-spontaneous, a lot of times people correlate that with happening fast or slow. That's not accurate. That's the wrong definition of spontaneous. Spontaneous means it can happen on its own. Non-spontaneous means it cannot happen without some help, in essence. So if we say somebody has a spontaneous personality, it's probably somebody that's always ready to go out and go to a party or do something, and nobody else has to tell them to do that. They're the extrovert that is then dragging the introverts to the party, for instance. Does that make sense, everybody? If somebody has a spontaneous personality. Spontaneous means it can happen on its own. Exergonic reactions can happen on their own. They're spontaneous because they don't require an input of energy, and in fact, they release energy. Endergonic reactions are non-spontaneous because you do not have this input of energy, this reaction would never occur. You have to have the input of energy for that reaction to take place. Questions about that? For that one, are anabolic chemical reactions then, are they favorable or unfavorable? Maybe I'll switch this. Are anabolic reactions, are they exergonic or endergonic? Based upon the definition I gave you earlier. Show of hands, you got to pick one. Are anabolic reactions, how many of you think that they are an example of exergonic reactions? All right, how many of you think they are endergonic reactions? All right, so they are going to be endergonic. Okay. Are they favorable or unfavorable then? Unfavorable. Would they be spontaneous or non-spontaneous? Non-spontaneous. Because remember, you can actually use those terms, endergonic, unfavorable, or non-spontaneous. Those terms are somewhat, again, interchangeable. They're describing the same thing about the chemical reactions. Catabolic reactions, are they favorable or unfavorable? Favorable. Are they exergonic or endergonic? They're going to be exergonic as well then. Okay, and they're going to be spontaneous. Yeah. Nope, I do. Questions about that? Okay, so here I have a graph. And it's showing a chemical reaction. So I have A and B. Are A and B the reactants or the products? Yeah, those are going to be the reactants. It's going to go through a chemical reaction. The arrow always denotes a chemical reaction taking place. And then C and D are going to be the products of that chemical reaction. One thing that I want to point out, a lot of times A and B will go through what is called, these again, the A and B are the reactants. a lot of times they go through what is called a transition state. You do not need to worry about this, but the transition state here is called AB prime. So they all reactants get chemically modified. Sometimes they get modified into this very short-lived transition state where their structure has changed, but they haven't fully formed the products yet. But eventually then they form the products then of C and D. So sometimes there's some initial small changes into a transition state called AB prime. Don't worry about that too much, okay? But this is what's being shown there. This is the progress of the chemical reaction, again, on the x-axis versus free energy on the y-axis. So as A and B and the reactants, they go and get converted into a very short-lived transition state. And as they then proceed through the chemical reaction, they get converted into the products of C and D. Is this graph showing a exergonic reaction or a endergonic reaction? Again, you guys got to pick one, and we're going to do show of hands. So how many of you think that this is depicting an exergonic reaction? Okay, how about endergonic? All right, that is going to be an exergonic reaction. It's always about where it starts and where it ends, and you ignore the middle when you're trying to figure this out. The reactants are at a higher energy state than the products. Okay? And the delta G of a chemical reaction, the delta G is always measured from where you start to where you end. So that value from the energetic state of the reactants and the products determines the delta G. Because remember, the delta G is just simply, delta G is simply equal to, again, the energy state of the products minus the energy state of the reactants. Where do you start? Where did you finish versus where did you start? It doesn't matter what's in the middle when calculating the delta G value. Questions about that? However, we are going to talk about what's happening in the middle. So, what is this then? This is the progress of the reaction, and you can see the energy state goes up at first and then comes back down. So anybody know what this initial kind of energy hill is called? Where you actually have to put a little bit of energy in to get to the transition state, and then you come back downhill and form the products. This is, what is that called? This is called activation energy. And sometimes it's also called an activation energy barrier as well. Activation energy. This is an initial input of energy necessary to start a chemical reaction. So activation energy is an initial energy input, or it's an amount of energy that is necessary to start the process of converting the reactants into the products. And that initial input of energy will take the reactants to a transition state. Once they get to a certain point, though, that transition state will get converted into then the products. Different types of chemical reactions need different amounts of activation energy. But you have to do that initial energy input or you don't get that reaction to occur. And by the way, you have activation energy needed in both endergonic and also exergonic reactions. All chemical reactions needs a little bit of activation energy. Now, the thing with activation energy, you have to put in a little bit of energy. So we went up in energy, but then you get that energy back out. Okay? As the reaction finishes, you get that energy back. And so the only change in the energetic state over time is then going to be that delta G value. Okay? Because you have to put in a little bit of energy to get to here, then you get that energy back, plus a little bit more out in the case of a exergonic reaction. Questions about that? Now, why do you need activation energy? If I had A was a molecule was sitting on this table, and if I had B was a molecule sitting near the back of the room, could they undergo a chemical reaction? No, they're on completely different side of the rooms. For things to go chemically react, the molecules have to be, well, touching. They have to be right next to one another, and they have to be right next to one another on a molecular scale. Okay? Things can't undergo reactions. If I have a molecule sitting in this room and a molecule sitting over in the university center, they're obviously not going to undergo a reaction. Okay? But not only that, even if they're just doing this, you guys know what absolute zero is? What's absolute zero? No heat energy whatsoever. Do molecules move at absolute zero? No, they just stay here. So say I have a molecule very close, molecule A, very close to B. Even if they're very close, if they're at absolute zero, can they undergo a chemical reaction? No, because even if they're close, they can't move to touch one another, which means they can't react. So with absolute zero, there's no energy. With no energy, there's no activation energy, and they can't react, for instance. So what I mean exactly by activation energy? It could be a couple different things. For instance, A and B to react have to come very close together. Have to come very, very close together. And the more they're moving around, guess what? The more likely they are to bump into one another. So if they're moving, they're more likely to hit, they're more likely to react. So one version of then activation energy is just simply kinetic temperature heat energy. Molecules, if they're cold, they move less. Cold is the absence of kinetic energy. If molecules are hotter, they have more kinetic energy, and kinetic energy is the energy of movement. So if they're hotter, there's more heat energy, they're moving around more, and guess what? They're more likely to bump into one another and more react. So one version of then activation energy is just simple kinetic heat energy. The more the molecules are moving around, the more likely they are to bump into one another, the more likely they are to react then. Questions about that? So temperature can be then a type of activation energy that is needed for these chemical reactions to occur. Another version, though, of activation energy is, for instance, you can mix hydrogen gas and oxygen gas. Hydrogen gas is H2, oxygen gas is O2. They love to interact and then react and form water over the course of a chemical reaction. So you can get the formation of water from hydrogen gas and oxygen gas. But if I mix hydrogen gas at room temperature and I mix oxygen gas at room temperature, they're going to bump into one another all the time, but they're still not going to react at room temperature because there's not enough activation energy at room temperatures, in essence. Why won't they react even if they're bumping into one another? Even if it's energetically very, very favorable reaction? Because hydrogen gas, H2 gas, is a hydrogen joined together onto another hydrogen by a covalent bond. Are covalent bonds weak or are they strong? Strong. Oxygen gas is an oxygen being held onto another oxygen by actually a double covalent bond. Two covalent bonds hold those together. Those are strong bonds. Remember, when you want to then make a reaction happen, you have to have the reactants go to a transition state. In this case, the transition state would be to the hydrogen separated and the oxygen separated. So one of the first things you have to do in this chemical reaction is you have to break the chemical bonds of the reactants. And then you have to reform the new chemical bonds of the products. But the transition state is that hydrogen gas broken apart, separate hydrogens. It's that oxygen gas broken apart. So instead of two oxygens covalently bonded, the oxygens are separated apart. Then they reform the new bonding patterns of the water molecule where the hydrogens bond with the oxygen. So there's that transition state again. But it takes energy to break the existing bonds of the reactants. So you need energy to break the bonds in the reactants, break those apart so that you can then form the new bonds in the products. Questions about that? And it takes sometimes quite a bit of energy to break covalent bonds. If you mix oxygen gas and hydrogen gas, again, at room temperature, nothing's going to happen. But just light a match and it explodes. It actually makes a giant fireball, but then what comes out of the fireball is water. As the hydrogen gas and oxygen gas gets broken apart, the heat from the match is enough to start the process. And the process is a very exergonic process as well. Okay. Now, this is then also where enzymes come into play inside of cells. Enzymes. And you guys should know this term and this definition. Enzymes are biologically produced molecules. Usually they're proteins, but sometimes they're RNA molecules. Most enzymes are proteins, but there are some RNA molecules that also function as then enzymes, like 16S rRNA or 23S rRNA, for instance, as some examples. Those are enzymatic RNAs. Enzymes are biologically produced molecules that then serve as catalysts. And a catalyst is any compound that speeds up chemical reactions. But enzymes are, not all catalysts are enzymes. There are several things that can act as catalysts that are just out in nature, but they're not produced by living organisms. Enzymes are specifically things that are produced and made by living organisms, and then they speed up chemical reactions. Okay? All enzymes speed up chemical reactions by lowering activation energy needs. So this right here is a chemical reaction, exergonic or endergonic? Exergonic. But this is the chemical reaction in blue without an enzyme versus this is the chemical reaction in red with an enzyme. The starting energy state did not change. The final energy state did not change. The delta G did not change. The only thing that changes in the presence of in the presence of an enzyme is how much activation energy is needed. Without an enzyme, you need a lot of activation energy to start this chemical reaction. With an enzyme, you need less activation energy to get that reaction. But I also said the activation energy is often a barrier. It's sometimes called an activation energy barrier. The more activation energy you need, the more of a barrier to a chemical reaction. And that means that chemical reaction happens slower. If you need less activation energy, there's less of a barrier, and that reaction happens much more readily, and therefore also much, much faster. And in fact, enzymes, just by lowering the activation energy barrier, will speed up chemical reactions anywhere from 10 to the 8th to 10 to the 20th times faster. Enzymes speed up chemical reactions a lot just by lowering the activation energy needs. Very, very fast. Enzymes are the dominant factor inside of living organisms as to the speed of their chemical reactions. And that's why, for instance, if proteins are denaturing, enzymes are denaturing inside of cells, well, that cell's probably dead soon because the enzymes are so important to it. How do enzymes lower activation energy needs? They do this in a few different ways. One, binding of the enzyme increases local substrate concentrations. Again, substrate, I can use that term interchangeably with reactants. Remember, substrates, reactants are the same way. This is basically just a fancy way of me saying they help bring reactants together. If you're by what I mean by increasing the local concentrations, just means they bring them close together. very close together so that they are bumping into one another so that they can react. The other thing they do is they ensure substrates are oriented properly to react. So typically, when you have a reactant interacting with another reactant, typically the reactants have what are called functional groups, like hydroxyl groups. So sugars. Hydroxyl group on one sugar will interact with a hydroxyl group on another sugar, and then that joins the sugars together into, say, like a disaccharide sugar. But if my sugar is one way is like this, and my other sugar is like this, are they going to react if they bump into one another? No, because my functional groups are on the wrong sides. If instead my sugars are brought together like this, they will react. So when I say oriented properly, I mean they bring the substrates close together, the reactants close together, but they also make sure the reactants are brought together in the right way, the right correct orientation, so that they are going to undergo a chemical reaction. And it just kind of looks like this, typically in the enzyme. So if this is my enzyme, at some place in the enzyme is what is called an active site. This is the portion of an enzyme that directly interacts with reactants, directly touches them. So the substrates or the reactants are going to fit into these active sites. Now here on this diagram, my active site, you might notice that they fit shape-wise with the shape of the enzyme is going to fit together like puzzle pieces with the shape of the substrate. That's not on accident here. The enzyme shape helps its function. And it also dictates what substrates a reactant is going to bind to. So substrate 1 comes into this active site binding pocket. Substrate 2 comes into this other active site binding pocket. But it brings it in a way that they're going to get very close together and in the correct orientation so that they're going to undergo a chemical reaction and then make products. What this also means is because the enzyme brings them together, you need less kinetic energy for this reaction to occur, or less activation energy for this reaction to occur. The other thing that happens with enzymes, the other way that they lower activation energy is something called induced fit. Will then put physical or electrical strain on substrate bonds. All right, so what do I mean by this? Here's an example of this point on the next slide. Enzymes are perfectly shaped to bind onto their substrates. A consequence of this, enzymes only bind to their specific substrates. And really, they only speed up one kind of chemical reaction. They always speed up the exact same chemical reaction. It's a shape always correlates to function. So enzymes are reaction and substrate specific. But if this is my enzyme here, this is my substrate in purple. When the substrate comes into the enzyme, it binds into the little active site binding pocket right here. And you can see it's shaped to fit together. But when that happens, the enzyme changes shape and it will wrap around the substrate. So here's my substrate. It's coming into my enzyme, and the enzyme does this. But then it starts to squeeze the substrate. It starts to physically stretch the substrate or electrically strain the substrate. It puts like negative charges next to other negative charges. Negative charges don't like to be next to one another. They repel one another. What this does, though, is takes all the bonds in the substrate and stresses them. It stretches them apart. So say there's a covalent bond holding together the oxygens. It starts to stretch those covalent bonds apart. And do you think you're weakening those covalent bonds then when you're stretching them? Yeah. By stretching the bonds of the reactants, you weaken them and you make them easier to disrupt. That makes it easier to then break those bonds than to make also the products. Okay, questions about that? Okay. The other thing to keep in mind with enzymes is they participate in a reaction, but they are not permanently changed by the reaction. Enzymes can get reused and recycled. So an enzyme can speed up a version of a chemical reaction one time, then it can speed up a second version of that chemical reaction again a second time, then it can speed up a third version of that chemical reaction a third time. They can get reused and recycled multiple times. And again, this just looks, this point right here, this recycling point, it kind of just looks like this. So the enzyme, here's the enzyme. It's got the active site, the binding pocket. Yeah. We're going to come back to that. So the other thing is that the enzyme, here's the binding pocket for the enzyme or the substrate. These substrates come into the active site. When the enzyme binds to its substrates, it changes shape. So you see my enzyme has changed shape, what's called induced fit. That'll put physical or electrical stress on those substrates. It helps bring those substrates together, in the right orientation, and all of that lowers the activation energy. Those get converted into products, the products get released, but the enzyme will flip back to its original shape. And then it could speed up a new version of that chemical reaction. So that enzyme can get reused multiple times and recycled. Questions about that? Again, in the end, enzymes accelerate chemical reactions anywhere from 10 to the 8th to 10 to the 20th times faster than without the enzyme. But all they do is lower activation energy needs. They do not supply energy itself. They just make it so you need less energy. Okay? Questions about that? Okay. One other thing to keep in mind with chemical reactions, all chemical reactions are reversible. So I've been showing chemical reactions like they just go one way. It actually turns out that chemical reactions can have a forward direction, but also a reverse direction. So if A and B can undergo a chemical reaction to make the products of C and D, you can also do the reverse reaction where C and D undergo a reverse reaction to make A and B. So all chemical reactions are reversible. You can go the forward direction, the reverse direction. And sometimes reactions reach what's called chemical equilibrium. Chemical equilibrium is when the rate of the forward reaction is equal to the rate of the reverse reaction. So the forward reaction is going at the same rate as the reverse reaction. And that might look a little bit like this. Chemical equilibrium is, again, when that rate, how often you go forward, is equal to how often you go reverse. But you might have different concentrations of reactants and products to when you're at chemical equilibrium. The other thing is one of the directions is going to be favorable or exergonic. The other reaction, the reverse reaction, is typically then going to be unfavorable or endergonic. So one direction is going to be exergonic. The opposite direction is going to be endergonic when you're going forward or reverse in these chemical reactions as well. Okay. Some reactions are a little harder to reverse than others. They might require more energy to reverse. The cell can also control what kind of enzymes or what kind of reactions they're doing with enzymes. So for instance, you can couple unfavorable reactions. Again, unfavorable is endergonic. You can couple them to very favorable exergonic reactions using enzymes. This is called energy coupling. You can use an enzyme. This might be, for instance, a exergonic reaction. This gives off free energy. The enzyme will then couple that to an endergonic reaction, and the free energy is used to drive that particular reaction. Questions about that? I know I'm kind of short on time. I'm going to do just two slides to finish up the storyline here real quick. Anabolic reactions, again, are energy consuming. Catabolic reactions are energy producing. So catabolic reactions will reduce energy, and they'll harvest that energy and store it in the form of energy intermediates. Those energy intermediates then go into anabolic reactions to drive those. Then that's something else we talk about. When we talk about energy intermediates, that's where I'm going to pick up on Friday. But there are three main energy intermediates that all cell uses. This is the last slide for today. There's three main ways that all cellular life stores energy. Bacteria, archaea, plants, us, animals. All store energy using three main energy intermediates: proton motive force, or PMF for short, NADH, and ATP. And we're going to pick up on Friday talking about these three energy intermediates, how each stores energy, and then we're going to start talking about how they get made and how they get used over the next couple weeks then.